everyone, and once again I welcome you all to the biology class. So students, we are discussing about the chapter that is a transport in plants. In the transport in plant, we have already discussed about the active transport, and we were discussing about the passive transport. In the passive transport, we have discussed all those that how it actually happens, what are the different kind of procedures that happens in this passive transport, and we have discussed about the diffusion process, right? Now, so in the previous class, I was talking about the osmosis, right? In the osmosis, I have discussed about the different kind of solution. We have discussed about the hypertonic solution, hypotonic and isotonic solution. Let's revise that and continue that. So, we were talking about the osmosis. Now students, tell me one thing, what is osmosis? You must have seen the previous lecture. That means a movement of the water from a region of a higher concentration to the region of lower concentration, right? So, that is called as a osmosis, right? When we talk about the osmosis, this basically transfer from a semi-permeable membrane. That is a movement of water is having from a, you can say, the higher concentration of the water to the lower concentration of water, and this whole process goes on. So, in this I was talking about the three basic type of solution. Hope you remember that. The first basic type that I was talking about was a hypertonic solution. Hypertonic solution. Hypertonic solution is that solution which is having a more concentration of solute and low concentration of solvent, that is called as a hypertonic solution. Another that I was talking about, that is a hypotonic solution. Now, hypotonic solution is that solution that is having a less amount of the solute and more amount of the solvent, that is called as a hypotonic solution. We cannot say that this particular solution is hypotonic, this is a hypotonic when it is present in a one medium, or you can say when we have only one solution. We cannot say that. It is actually a comparative property. Whenever we compare one of the solution with another solution, this property come into existence, right? Let's talk about the third type called as isotonic solution. Isotonic solutions are those solutions which are having a same osmotic concentration with respect to each other, right? So, these are the different type of the solution, hyper, hypo and the isotonic solution. Now, let's understand this different type of solution with the help of a diagram. Now, look at this side. Suppose I have a membrane like this, right? And this is how they are separated from one another, right? In the center, this blue color, this is a semi-permeable membrane. Semi-permeable membrane. When we talk about the semi-permeable membrane, that means it allows only a solute to pass through it. It doesn't allow the solvent to pass through it. This is a characteristic feature. Suppose at one side I am having one kind of solution. Let's talk about I have hypertonic solution. Hypertonic solution. Right? Hypertonic solution, it is present over here. That means it is having a more concentration of solute, more of this red color that I am drawing. Suppose these are the solutes, right? Right? And in this I have a very less amount of the solvent that I am drawing with the help of this white color, right? This white color, they are what? They are solvent. right? This is an hypertonic solution. Let's talk about the another solution. Suppose in that the number of the solutes, they are very less, right? And the number of the solvents, they are more. Very large in number, and that solution will be called as what? That solution will be called as a hypotonic solution. Hypotonic solution. Right. Now, understand this, because we have discussed about the three type of solution. Now what will happen? Because solvent are more in this, solvent are less in this, and I've told you that there is a movement of the solvent from higher concentration to the lower concentration. That means a movement will occur in this direction. Water will move like this, or the solvent will move like this, until or unless a balance is maintained between them, right? So this is a diagram which shows that how this movement occur across the semi-permeable membrane. That means it's a diagram of osmosis. Hope everything is clear to you. Now, come at this side. Let's talk about the type of osmosis. Let's discuss the type of osmosis. Now, type of osmosis, that means what actually is happening. Sometimes the movement of water occur from outside to inside in the cell. Sometimes what happens, the movement of water, they occur from inside to outside. That depend upon the surrounding medium, what is present in the surrounding medium. So on the basis of that, we have classified the osmosis into the two categories. The first category is called as endoosmosis, and the second category is called as exoosmosis. Now, let's discuss these two procedures. So, as the name indicates, the endoosmosis, that means suppose a cell is placed in a medium, in a water. Now, what will happen? There will be a movement of the water. From where? From outside to inside. That means from region of a higher concentration of water to the region of the lower concentration of the water. This is what we call it as a endoosmosis. So, write in this, movement of water will be from outside to inside of cell, right? And when this will happen? When cell will be placed in hypotonic solution. Hypotonic solution, right? Now, listen to this, how it will happen. Suppose I have one cell, right? And that cell is placed in a, suppose this is a beaker, and I have drawn only a single cell, right? Actually, this doesn't exist like this, and I have placed some water, or you can say hypertonic solution all around. Sorry, hypotonic solution. Hypotonic solution I have. This is a hypotonic. And this is a cell, and the cell will be having the protoplasm, cell will be having the different organelles, cell will be having the organic substances which will be helpful for building of the cell. In this, the cell, the solution will be considered as a hypertonic. So, they will become hypertonic. Now listen to this very carefully. When we talk about the endoosmosis, the hypotonic solution is present outside. That means more water is present outside. In the cell there is less water. Now what will happen? The movement of the water will occur from outside to inside like this. At this particular stage, what will happen? The cell will become turgid. Cell will become turgid. Right? This is a first example of endoosmosis. Now, remember this word turgid because in this class only I will be talking about the turgor pressure. So just remember, now what will happen? As the water has entered into it, now the size of this cell will be more. They will accumulate more and more amount of the water and the cell size will increase. Right? Now let's talk about the second process called as a exoosmosis. It is opposite of the endoosmosis. In the endoosmosis, the water has moved into the cell. Now the cell has become turgid. Now what will happen in the exoosmosis? Exo means outside. Water will move outside. So what can I write over here? That means a movement of water from inside to outside of cell, right? When cell is placed in hypertonic solution. Right? Now, again, let's draw this diagram. So, what I have one flask. In that flask, I have placed one cell. Suppose this is the cell. After some time, suppose I have filled the solution over here, and the concentration of that solution, the concentration of solute in that solution is more. That means this solution will be called as hypertonic solution. Hypertonic solution, and this is called as a hypotonic solution. Now, this is opposite of that. Now, water will go from a region of a higher concentration to the region of lower concentration. So, water will move like this. As the water will move like this, there will be shrinking of cell. Now, the cell shape will be like this. Right? This I can call it as a shrinking of cell. Right? And this shrinking of cell is also called as a flaccid. The cell will become flaccid. Flaccid. That is also called as a flaccidity. Water has moved outside, and we know that all the cell membrane, they are of semi-permeable membrane. Or I have used, that is basically semi-permeable as well as selective permeable. So, this is how we can explain the exoosmosis, right? Now, listen to this very carefully. Initially, endoosmosis I have explained, where the movement of water has occurred inside the cell, that is from the hypotonic to the hypertonic solution. So, whenever any questions like this is asked in your NEET exam or any of the exam which you are appearing for, then do remember one key thing. That means movement of water occur from hypotonic to hypertonic solution. Just remember this line, then you will be able to solve all those questions that are related to the osmosis or that are related like what will be the condition of the cell, well the cell will become plasmolized, well cell will show endoosmosis, exoosmosis. So just remember this very carefully, right? This was the turgid and this is a flaccid cell, right? Now come at that side, let's talk about the example. Let's talk about that how this osmosis occur. So, suppose this is a stand, let's do an experiment. This experiment is mentioned in your books. So, just remember that how this happens. This is a stand, right? On this stand, I have one beaker over here. Right? On this beaker, I have hanged one this is a stand of the stand, and in this stand I have, suppose I have this pipette-like structure. In this pipette-like structure, I have filled a glucose solution, or you can say a sugar solution. And this sugar solution, I have bind this sugar solution with the help of a cloth. And that cloth, I will call it as a semi-permeable membrane. Right? And this is filled with the sugar solution, is present over here. This is a beaker, and in this beaker I have water. Right? And this is the water. Now listen to this very carefully. I have told you that the osmosis occur from a semi-permeable membrane, and this I am labeling it as a semi-permeable membrane. Semi-permeable membrane. Now I have told you that the movement of the water occur from a hypotonic solution to the hypertonic solution. This sugar solution as compared to the water solution, this is a hypertonic solution. Right? Whereas this water is hypotonic. Right? Now what will happen? The water will migrate from this to this semi-permeable membrane. What will happen? The water level will rise. This is the initial water level. This I am labeling it as an initial level. Right? And after this entering of the water from the outside, some of the water will enter into it, due to which there is a raise in the water level. So after some time, there will be a final level attained, because the migration has occurred, the movement of the water has occurred from the outside to inside of this, you can say the sugar solution, because this is a hypotonic and this is a hypertonic. Now the raise in this water level can be seen. Right? So this is called as a osmosis. Right? So in this case also the movement of the water has occurred. Movement of water has occurred from the hypotonic solution to the hypertonic solution, that is from the outside to inside. Initially the level was this thing, right? This level was there and now we have this level. This is called as a final level, and this whole process is termed as osmosis, right? And we have seen the semi-permeable membrane is also present over here. So students, note down this. So students, hope you have noted down about this whole process, the experiment which is related to the osmosis, right? I hope you can write all this in your words. Now, let's talk about the osmotic pressure. That what exactly the pressure that is exerted by that solution due to which it passes from a semi-permeable membrane. Let's talk about that. Osmotic pressure. Now, again listen to this. When we are talking about the osmotic pressure, that means it is that pressure which is exerted by the solution when it is placed in close contact by semi-permeable membrane with pure water. That actual pressure that exists in a solution is called as osmotic pressure. When we talk about a plant cell, osmotic pressure of that plant cell is around 4 to 5 atmosphere, right? So when we talk that how we can, the symbol of osmotic pressure is like this, pi, light on. So we are talking about the osmotic pressure. That means a pressure exerted by, or the actual pressure that develops in a solution, actual pressure that develops in a solution when it is placed in pure water separated by semi-permeable membrane. Separated by semi-permeable membrane, this is called as a osmotic pressure, right? Denoted by the symbol pi. When we talk about the osmotic pressure, let's take an example, that osmotic pressure of a plant cell. Right, when this plant cell is placed near a pure water, the osmotic pressure is around 4 to 5 atmosphere. 4 to 5 atmosphere, right? This is an osmotic pressure that actually help in osmosis, right? So, what exactly the factors that determine the osmotic pressure? The one of the factor is the concentration of the solution, right? Osmotic pressure depend upon the following factors. The first is this is a concentration of solution. Next, it depends upon the ionization rate of that solution. And ionization and hydration. Right? Now, with this we have completed the osmosis. Now let's talk about what are the functions of osmosis, what actually they do. Let's talk about the osmosis functions or role of osmosis. one of the important factor, one of the important use of this osmosis, one of the important role of this osmosis is that they maintain the turgidity of a cell. Right? They help in opening and the closing of the stomata. So, whenever any questions like this is asked, you have to remember the thing is clear, this thing should be clear in your mind, that this osmosis, they help in opening and the closing of the stomata. And we know that this stomata closing and opening, they determine the transpiration rate. So, the first example, they maintain turgidity of cell, right. Second, I have told you that they help in opening and closing of stomata. Now, another important process, another important role is that whenever a plant is placed in a soil, suppose a plant is there in the soil, the amount of the water that is it absorbs, that is because of the osmosis. So, the function of it is, that is absorption in absorption of water, right, from roots. Right, this is because of the osmosis. Another important role of this osmosis is that osmosis not only help in all this process, instead they also help in the movement of the plant and the plant parts. Movement of plants and plant parts. Right. So, these are the certain functions: maintain turgidity, opening and the closing of the stomata, absorption of the water from the roots, and the fourth one, movement of the plant and the plant parts. These are the certain important uses of osmosis. Right. Now, let us talk about the another important process called as plasmolysis. Let us discuss about the plasmolysis. Now, what is plasmolysis? Plasmolysis is a movement of the water. Suppose I have done an experiment. In that experiment, I have placed a plant cell in a water, and that water is a hypertonic solution. So that what will happen? There will be exoosmosis. The movement of the water will occur from inside of the cell to the outside of the cell. This is called as exoosmosis, and this is also called as, you can say, the plasmolysis. Right. So, outermost solution that we have, that is a hypertonic, right. You have to remember, do not get confused between these words, right. So, when plant cell placed in water, not water, the hypertonic solution. Hypertonic, they are placed in hypertonic solution. Now what will happen? Movement of water from inside of cell to outside of cell occur, right. Cell protoplasm will shrink, right. And when this plasmolysis keeps on going, then ultimate stage, the last stage that will come, due to which the plant cell will die. If this process keeps on going, the plant cell will die. plant cell will die, right? This is called as a plasmolysis. Now you have to remember the exact definition. When plant cells, they are placed in hypertonic solution. Hypertonic means outside the solution, sorry, actually it is placed in, when the plant cell is placed in a hypertonic solution. The movement of water from the inside to the outside will occur, that will lead to the cell shrink, and this process keeps on going, right? So students, note down this. So students, this is a plasmolysis. That means when a plant cell, this is placed in a hypotonic, this is, it is a hypotonic, because we know that the movement of the water, they occur from hypertonic to the hypotonic solution. The movement of the water from inside of the cell to the outside of the cell will occur. The cell protoplasm will shrink. If this process keeps on going, the cell will ultimately lead to the death. This is called as a plasmolysis. When we talk about the types of plasmolysis, let's discuss that. So, let's discuss the types of plasmolysis. Right. There are three types of plasmolysis. First is called as a limited plasmolysis. Limited plasmolysis. That means suppose one of the plant cell, this is kept in a hypotonic solution. Movement of the water will occur from inside of the cell to the outside of the cell. Now what will happen? There will be less of the turgor pressure, that is will be exerted to the wall, right? The turgor pressure at that particular time will be 0. This is called as a limited plasmolysis. Right. Let's first write about the three types. The limited plasmolysis. Next we have is an incipient plasmolysis. Third is evident plasmolysis. These are the three types. Now come at this side so that we can do all these type of plasmolysis with the help of a diagram. Let's do this now. Now, suppose I have one cell. The first we have is the limited plasmolysis. Let's talk about the limited plasmolysis. Suppose I have one plant cell. Let's draw the structure of the plant cell. So this is how a plant cell look like. This is how a plant cell look like. In the plant cell, this is the outermost cell wall. Right? Outermost cell wall, along with this, the protoplasm will be present over here. That are separated from the cell wall by the plasma membrane. By cell membrane, sorry. Right? This is our protoplasm. Now, what is happening? I have placed this cell in hypotonic solution. Now what will happen? Normally, a turgor pressure exists between this protoplasm and the cell membrane. This turgor pressure means that pressure which a protoplasm apply on the cell membrane. Right? In this case, the turgor pressure will be 0. This is a cell placed in hypotonic. Just now we have placed it. Now, in this case, the turgor pressure will be 0. You have to remember this. This is called as a limited plasmolysis, right? It is recently placed, pressure has decreased, that a protoplasm applies on the cell wall, right? And another cell organelles, they are also present like chloroplast, they have chloroplast, mitochondria, etc. Right? Now let's talk about the second type, which is called as a incipient plasmolysis. Now, focus on this diagram. Now, protoplasm is like this. Now, it is placed in the hypotonic solution, and after some time, we will able to see that the cell wall, they will remain like this, right? Right? But this protoplasm has shrinked. Protoplasm has shrinked. Now the protoplasm has shrinked. Now even what has happened? The protoplasm shrinked, even the vacuole has shrinked. Vacuole has shrinked, right? So these are the two processes that has already been occurred. The first, the protoplasm shrinked, and next is the vacuole shrinked. The both are shrinked, right? This is how our protoplasm look like. This is between a gap between the plasmolysis initial. This is a plasmolysis has just started. Now plasmolysis is going on, right? The shrinkage is occurring. Now let's talk about the third type that I have written, that is called as evident plasmolysis. Evident plasmolysis. Similarly, the cell we have a cell over here, plant cell, and this plant cell, the cell wall remain like this. Right? Now, this protoplasm will be reduced to a very small size. Protoplasm shrinked. It is properly shrinked, right? Now, in this case, I forgot to write over here about the Turgor pressure. Initially, it was considered 0. Now, the Turgor pressure will be negative, right? In this case, the Turgor pressure will be more negative. Turgor pressure more negative. More negative, right? Now, see, can you see how this plasmolysis occur? This is one after the another. Cell wall, the protoplasm it keeps on shrinking. Then ultimately when this position will be attained, there will be cell death. Right? Now, listen to this again carefully. We are talking about the plasmolysis. That means I have placed this cell into the hypotonic solution. Hypotonic solution is there. Movement of water will occur from inside to the outside. Movement of the water will occur. Now, what will happen? The protoplasm will shrink. It has started shrinking. That's why it is called as a limited plasmolysis. The Turgor pressure, that pressure that a protoplasm exists, a protoplasm they basically forces this cell wall, right? But in this case, the Turgor pressure will be 0 because it has started shrinking, right? Even the vacuole size, they have started shrinking. The vacuole, this is a characteristic feature of the plant cell. Now, let's talk about the second, which is called as an incipient plasmolysis. Protoplasm shrinked, right? Vacuole is also shrinked. Turgor pressure is negative. Over this case, incipient plasmolysis, that is after some time of this plasmolysis. You can say after so many hours, what will happen? The protoplasm will completely shrink. Turgor pressure will be more negative. and the cell death occur. Ultimately, it leads to the cell death. Right, students? So, these are the different type of the plasmolysis. Along with the different types of the plasmolysis, we have certain examples as well. Have you ever heard of imbibition? Imbibition is also a procedure by which the cell, or you can say some solid substances, for example, you all must have grains in your houses, or you must have used grams, right? And whenever you soak those grams, in the next morning you can see that those grams, those you can say in like pulses that you used to soak, that has swollen, that has increased their total surface area. When it has increased their surface area, and during the whole process of imbibition, you can see that water has become little hot, right? This is called as a imbibition. Right? We will be talking about the imbibition in the next lecture. Till then, understand this plasmolysis very carefully, right? Watch this lecture. I am again telling you that definitely 1 or 2 questions will be asked from this chapter only. So, this whole chapter seems easy, but sometimes some of the students can get confused between them. So, students, this is all about the today's lecture. We will meet in the next lecture.